Hello friends, meet Tommy, a little lion lap rescue rabbit with a very unique coat pattern. As you can see, little Tommy has a mostly white body with some patches of gray throughout her body, particularly concentrated around her ears, eyes, and nose, but there are a few patches on her back as well. Today, we want to know what her potential genotype is for her coat coloring, given this adorable and unique phenotype. How would we do this? Let's talk about it. What determines a rabbit's coloring? Genetics. Coat color is based on several different genes that control color base, color density, pattern, extension of dark pigment, spotting, and silvering. Keep in mind that there is still much that is unknown about the rabbit genome, but today we're going to discuss specific aspects of the rabbit genome that we do know and how they interact to form what we visually see, also known as the rabbit's phenotype. Now, there are currently 7 different known coat color genes in the rabbit genome, and those that code for coat color and pattern are our A, B, C, D, E, spotting, and silvering genes. Let's learn more about each gene and how genotype influences phenotype. Brief disclaimer though, in this unit we will be focusing on the 7 known genes that have been confirmed to affect rabbit coat color. But there is still much unknown about the rabbit genome, and there are so many genes whose functions have not been identified or clarified yet. So we're just going to focus with what we know. So, starting with the B gene, because the B gene controls the coat color base. The B gene, specifically known as TYRP1 gene, controls the coat color base. The B gene has 2 alleles, big B and little b. This gene demonstrates complete dominance, with the big B allele being dominant to the recessive B little b allele. The big B allele produces a black color base, and a homozygous little b genotype produces a chocolate or brown color base. The big B allele is considered to be the wild type allele. The D gene. The D gene controls the density of the coat color. The D gene is specifically or scientifically known as MLPH, or melanophilin gene, and it controls the density of coat color. The D gene also has 2 alleles, big D and little d. This gene also demonstrates complete dominance, with the big D allele being dominant to the recessive little d allele. The big D allele contributes to a dense coat color, meaning that the base coat color will appear according to the B gene genotype. The little d allele represents a dilute coat color, meaning this will affect the coat base on the B gene. When a rabbit has a dilute genotype, meaning it must be a homozygous little d, a black base will appear as a grayish color. And for some funny reason in the world of animals, we call gray blue. So a black base will appear as quote unquote blue, and a chocolate base will appear as what we call lilac. The D allele is considered the wild type gene, the capital or big D allele. So looking back at Tommy, for the B and D genes, given the gray coloration on her ears, tail, nose, and around the eyes, and gray is a dilute form of black, we can likely assume that Tommy has a dilution gene at play here, suggesting that we have a genotype of big B and something, and little t genotype for her. We cannot be sure of the second allele for the B gene simply because both big B and big B little b code for black. But we know at least one of the B alleles is likely at play here, and it has to be a big B. And then we can assume that it is a homozygous little d genotype because it's not dark black, it's dilute. She has that blue-gray coloring. Moving on to the A gene, which controls the color pattern of the coat. The A gene, scientifically known as ASIP, or agouti signaling protein gene, controls the color pattern of the coat. The A gene demonstrates complete dominance again, with the A allele being the most dominant and the little a allele being the most recessive. The A gene has 3 known alleles. The big A allele causes the agouti pattern. The AT allele causes a tan pattern. And the little a allele causes the self pattern. The agouti pattern is considered the wild type allele, which appears as a multicolored, dark and light speckled coat, which comes from each individual hair having bands of color, dark alternating with light. The belly of an agouti rabbit is light, often a cream color. The tan pattern appears as a dark coat with a lighter red or yellow belly and nose, where each hair is a solid color with no banding. The self pattern appears as the phenotype based on the other coat colored genes. Each hair is a solid color with no banding. So looking back at Tommy for the A gene, since she does not have an agouti nor tan pattern, it is likely a self pattern, leading to the genotype of little a, little a. Now the C gene. The C gene controls the coloring of the pattern. So the C gene, scientifically known as TYR or tyrosinase gene, controls the coloring of the coat. In order of descending dominance, the C gene has 5 alleles. So buckle up. We have big C. Then we have little c with CHD. Then little c with CHL. And then we have little CH and little c. The C allele, the big C allele, stands for full color, and the color phenotype is based on the D, B, and A genotypes. The CCHD allele stands for chinchilla dark and causes the replacement of what would be yellow-red pigments with white cream pigments. For example, in an individual with a big A and little CCHD genotype, the yellow band on the individual hairs from agouti is replaced by white, giving a chinchilla coat. The CHL, so the CHL allele, stands for chinchilla light, also called sable, and seems to lighten pigments overall, but keep the ears, nose, feet, and tail darker than the body. The existence of the CCHL allele and its relationship with the sable phenotype has been speculated, but not confirmed with DNA evidence. The CH allele stands for Himalayan and appears as a white coat with dark ears, nose, feet, and tail. Something in particular that's really cool about Himalayans and other animals akin to the Himalayan rabbit is that we believe that these individuals actually have an allele that codes for heat-sensitive enzymes. And so how it works is the enzyme catalyzes a step in the synthesis of the brown pigment melanin. And so in central body regions where metabolic heat is pervasive, right? In those central body regions where we have that metabolic heat, the enzyme is disabled due to that heat. So no melanin is produced, and therefore in those parts of the body, the fur is white. But in cooler body parts, the enzyme is still functional, and so melanin is produced, and thus the fur is tinted that darker brown color. And so this kind of shows the effect of temperature on the rabbit's phenotype that was ultimately influenced by the rabbit's genotype. And we actually were able to learn this by keeping an area that usually would be Lighter, if you shave down that area and then keep that area cool, it'll grow back dark. So pretty cool. Okay, moving on. The little c allele stands for albino and appears as a completely white coat. The little c genotype is epistatic to all other genotypes. So if a rabbit is little c, it will be albino regardless of the rest of the genotypes. The big C allele is considered to be the wild type allele. So looking back at Tommy for the C gene, her white body with darker features around her nose, ears, and tail area suggests an influence from the Himalayan allele, or CH. But this is a prime example of where we don't always have a full answer, right? Because Tommy does not have dark feet that would perfectly fit the Himalayan phenotype. But we can assume that there is some influence from this gene given the rest of her phenotype. So now let's look at the E gene, which controls the extension of dark pigment on the hair shafts. The E gene, scientifically known as MC1R or melanocortin 1 receptor gene, controls the extension of dark pigment. In part 1, we discussed the E gene a bit and only mentioned 2 alleles. However, the E gene is actually a little more complicated. In order of descending dominance, the E gene has actually 5 known alleles. We have ED, ES, E, EJ, and little e. The ED allele stands for full extension, and the color will be determined by the B and D genotypes, no matter what the A and C genotypes are, except for little c. However, keep in mind, ED, this allele is actually very uncommon. The ES allele stands for partial extension and appears as steel, where individual hairs are dark with either silver or gold tips. For the steel genotype to appear, the rabbit must also have a big A in their genotype. The big E allele stands for normal extension and appears as the phenotype that is based on the B, D, A, and C genotypes. So the E allele is considered the wild type allele. The EJ allele stands for different extension by hair shaft. Originally known as Japanese brindling, an EJ rabbit will be harlequin or tricolored. And the little e allele stands for no extension, and the little e rabbit appears as fawn, orange, or red. The E genotype determines if and how much red or yellow pigment will be in the coat. So looking back at Tommy for the E gene, Tommy has color on her extremities, so she likely has a normal extension. But since she is mostly white, the genotype could be big E, EJ, or big E, little e. Spotting and silvering. The spotting gene, scientifically known as KIT, K-I-T, gene, controls white spotting on the coat. What we think of as typical spotting, or a broken coat, occurs with a heterozygous genotype of EN, little en. The genotype big En appears almost all white. This phenotype is sometimes termed as a Charlie. The genotype little en appears as no white spotting. The En allele is considered the wild type allele. The silvering gene controls the silvering of the coat, where white hairs are interspersed in a darker coat over the whole body. The gene that controls silvering has not been identified yet, but sometimes silvering is observed in heterozygotes that are noted as big Si, little si, and sometimes not. Only a handful of breeds have the little Si allele, silver, silver fox, champagne, and so on, with varied degrees of silvering. The big SI allele is considered the wild type allele. Looking back at Tommy for spotting and silvering, since Tommy is mostly white with color on her extremities, such as her nose and ears, she's likely big EN, little en for the spotting pattern. And since she does not show any silvering, she would likely be big SI, big SI. Wild type. A wild type coat, just as a refresher, refers to the quote unquote original rabbit coloring, meaning prior to domestication and without mutation. The wild type genotype is big A, big A, big B, big B, big C, big C, big D, big D, big E, little en, big SI, big SI. With this combination of alleles, the rabbit would have a chestnut color. Each hair over the top of the rabbit will be banded with black and dark yellow, while the belly will be white. So moving forward, you are going to determine your rabbit's or a rabbit of your choice's genotype. Feel free to reference this lesson and the additional materials provided to help you accomplish this goal. And have fun!